All right, let's talk physiology of skeletal muscle fibers. Oh no, I didn't want to do that. All right, we've gone through all of this. Here, we've checked out all the anatomy basically in the last video. And here we are at the physiology. So here now we talk physiology. We know how a muscle fiber contracts, right? We saw the anatomy of the muscle and we checked all that out. If you didn't have that thorough idea and understanding from anatomy, we reviewed that there for you. So we know how a muscle fiber contracts, but what induces it to contract? But what induces it to contract? You'll see here, we'll go through these different processes, different steps. You can see we've got them down in here as well to get broken down into two different phases. And we'll go through everything here and we'll check all these different steps out and we'll correlate them to then this image that we have here as well, right? And we'll go through everything, check everything out step by step and look at all that good stuff. Excites me. So here we can go through and we talk now, in order for a muscle to contract, right? We're talking skeletal muscle is what we're looking at here. Okay, it must be activated, number one. It has to be activated. So here you can see, and we're going to see, has to be activated. So has to be activated, number one. It has to be stimulated, you could say, by a nerve ending so that a change in membrane potential occurs. So number one, in order for skeletal muscle to contract, number one, it has to be activated. Has to be stimulated by a nerve ending so that a change in membrane potential occurs. Next, it must generate and propagate an electrical current. It must generate and propagate an electrical current. And then number three, okay, so going back to number two, we can see here the whole picture. So when we look at this picture, number one, it says that this nerve ending has to create a change in membrane potential. So once that occurs, then here on the sarcolemma, this muscle has to be able to, as we said, generate and propagate an action potential along its sarcolemma. And then number three, step three, that action potential, the action potential is propagated along the sarcolemma. So has to be able to, number two, generate and propagate the electrical current. And number three, then, the action potential is actually going to get propagated along the sarcolemma. And then step four, you can see there has to be a short-lived increase. We have to have a short-lived increase in the intracellular calcium levels. Right? That's going to be, we said yesterday or in the last lecture video, we said that is the actual final trigger. That's the go signal I was saying yesterday in the last video. That's the final go signal for contraction. So let's go through and let's look at basically all these different steps now, one by one. So again, it has to be activated. That's going to happen at the neuromuscular junction. And then you'll see the second part is excitation, contraction, coupling has to occur there in relation to the muscle in order for us to have the muscle fully contracted. So here now, when we go through and we talk step one and step two, that's basically those activation steps, and they're going to take place at the neuromuscular junction, and they're going to set the stage, you can say, or you can say that they're going to initiate the other events. I mentioned to you some other events in relation to step three and step four. That I mentioned there, step three and step four are going to be linked to the electrical signal to contraction, and they're going to be called basically excitation, contraction, coupling. So the second part we see here. Now here we can see in this image basically both of those major or all four of those steps there, the two divisions we've got there, all being summarized into two major phases. So if we look at all of this in detail, again, now you see action potential arrives at the neuromuscular junction, acetylcholine gets released, and it's going to get released. It's going to bind to the receptors on the sarcolemma on the postsynaptic neurons membrane, postsynaptic cells membrane. Here, the postsynaptic cell is not a neuron like we saw before, but here it happens to be a muscle. So then we have to have a change in ion permeability. That ion permeability, when it changes on the sarcolemma, this is going to help create local changes in the voltage, causing depolarization to occur. So then these local depolarizations, they're now going to be known as end plate potentials. We studied before graded potentials. End plate potentials we're going to see are going to be exactly what graded potentials are. And then they're going to now here, these EPPs are going to ignite action potentials in the sarcolemma, just like we saw graded potentials did, right? We had two different types of graded potentials. The EPSPs that were created with they could create basically two types of signals, EPSPs or IPSPs, right? Which were hyperpolarizations versus depolarizations. So here then in phase 2, steps 3 and 4, you can see now once this action potential is ignited, that action potential has to be propagated across that entire sarcolemma. It has to dip down those T tubules as we mentioned, and then help to release calcium from the sarcoplasmic reticulum. That calcium then, you saw, it binds to, very good, I know you all knew that, troponin, and then helping for myosin binding sites to be exposed, the active sites on then the actin, allowing the myosin heads to bind then to their actin, allowing for contraction or cross-bridge formation to occur. So let's look at then all of these different events. So again, now the nerve stimulus and the events at that neuromuscular junction. So skeletal muscle stimulated by, first you have to get understood the somatic motor neurons. They're going to be stimulated by somatic motor neurons. Now, axons of the motor neurons travel from central nervous system via nerves to the skeletal muscle. Each axon forms several branches as it enters the muscle. Right? We said it's going to make its way into the central part and it's going to branch profusely from there. Each axon ending forms a neuromuscular junction with single muscle fibers, And usually only one per muscle fiber. So here we can actually see now all of the events taking place here. So let's look at the nerve stimulus and the events that are going to be taking place at the neuromuscular junction. So like I said, somatic motor neurons or motor neurons of the somatic voluntary nervous system, right, are going to be nerve cells that activate skeletal muscle fibers. These motor neurons, they reside in the brain and the spinal cord, but they're long, thread-like extensions called axons. Their long axons are going to travel. They travel bundled within nerves. They're going to travel to the muscle cells. And then, like I said, they're going to divide profusely as the nerve enters the muscle, giving off short branches that form a neuromuscular junction or an end plate with a single muscle fiber. That form a neuromuscular junction or an end plate. Hence the name end plate potentials, the EPPs we were talking a few seconds ago. So here you can see again then a neuromuscular junction. Neuromuscular junction, right? Neuro because of the nerve, muscular, muscle, junction where two things come together. So a neuromuscular junction, let's talk first. A neuromuscular junction is where the branches of an axon ending are going to meet, meets with a single muscle fiber, as we described. The axon terminal and the muscle fiber are really close. Okay, so here you can kind of see that there, but if you move in this picture, you can see right inside of here on the top, this nerve component and the muscle, that neuromuscular components, they look very close. So again, the axon terminal and the muscle fiber, they look like they're really close to each other. And that's exactly what's happening. They are really close. However, they do, and you can see that in the picture as well, remain separated. They're remaining separated, you can see, by this space. By this space. This space we see here is called a synaptic cleft. It's called a synaptic cleft. So when we talk synaptic cleft is a space. It's a space found between the axon terminal and the muscle fiber. A space between the axon terminal and the muscle fiber. It's filled with a gel-like extracellular substance. It is filled with a gel-like extracellular substance that is rich in glycoproteins and collagen fibers. Also appreciated here are going to be synaptic vesicles. These synaptic vesicles are found within the axon terminal. So they are in the axon terminal. And what they are small membranous sacs. They are small membranous sacs that contain neurotransmitters. The neurotransmitter here that they contain is acetylcholine. So synaptic vesicles are found within the axon terminals. You can see those here very nicely. A whole bunch of little synaptic vesicles. And here there's one synaptic vesicle that's fused, and one that's starting to fuse, and one that's been fused for a while. So the synaptic vesicles are found in the axon terminal. They're small membranous sacs that contain the neurotransmitter acetylcholine. So all these little dots you see here in green, those are acetylcholine neurotransmitters. Acetylcholine. Acetylcholine, we studied in the last section, is a neurotransmitter, and it attaches to acetylcholine receptors. It's going to attach to acetylcholine receptors that are on the sarcolemma of the muscle fiber. So here you can appreciate all of those various receptors that we can appreciate now on the sarcolemma. A whole bunch of ligand-gated receptors, chemical-gated receptors, right? The ligand is acetylcholine. So it's a neurotransmitter that attaches to acetylcholine receptors on the sarcolemma of the muscle fiber. Then you'll question, how does a motor neuron stimulate the skeletal muscle fiber? Well, here we can see now when a nerve impulse, when a nerve impulse reaches the end of an axon, the terminal releases acetylcholine. So look at the action potential arrives at the end of the axon terminal of the motor neuron. And you guys all know this. Voltage-gated calcium channels open up. Because again, this is the action potential, voltage. Allowing then these voltage-gated calcium channels to open up. When these voltage-gated calcium channels open up, calcium makes its way in. And what was the name of the protein we said that calcium is going to go into and bind to? Very good. I know you all got it. My heart tells me you all got it. Synaptotagmin. Calcium will bind with synaptotagmin, and that's going to give the signal for exocytoses of these vesicles to occur. So it gets into all of this, I think too much detail for you guys. It shows the snare proteins and all that. You don't need to know all that. Just cut it short to you're going to have exocytosis take place. So when exocytosis occurs, now you know these synaptic vesicles, I told you they're membranous sacs, made up of the same component the membrane's made up of, which is? Very good. I know you said phospholipid bilayer. So here you can see they diffuse with one another, allowing the neurotransmitter to make its way out. So the neurotransmitter, okay, the terminal releases acetylcholine into that synaptic cleft. Acetylcholine then is going to diffuse. Acetylcholine will diffuse. Acetylcholine is going to diffuse across the synaptic cleft, and it's going to bind to those ligand gated calcium channels. So it binds to those acetylcholine receptors on the postsynaptic membrane, or you can say on the membrane of the sarcolemma. So here you can see now, as soon as acetylcholine binds, so it diffuses across, right? Diffuses across. Diffuse is a key word. Across that synaptic cleft and binds to the receptors on the sarcolemma. Now, when acetylcholine binds to the receptors, this acetylcholine binding triggers electrical events. Here now, specifically speaking, that acetylcholine binding opens ion channels. Now, the opening of these ion channels allows, you have to understand, it allows simultaneous passage. You can see that up here. It allows for the simultaneous passage of sodium, obviously, into the muscle fiber and potassium out, right? They're going to go down their concentration gradients. Now, more sodium ions are going to enter than potassium ions are going to exit. And now when you have more sodium entering compared to potassium exiting, what this does, it produces a local change in that membrane. It produces a local change in the membrane in its potential, right? And that change in the membrane potential is what we're calling an end plate potential. So the binding of acetylcholine to the receptor helps to trigger, you can see, end plate potentials. So the binding of acetylcholine helps to trigger end plate potentials. Now we have acetylcholinesterase. Acetylcholinesterase is an enzyme that's located in the synaptic cleft. It is an enzyme that is located in the synaptic cleft. And what acetylcholinesterase, I know you're all yelling at me, telling me it's an enzyme. I've taught you that suffix there, ase, is an enzyme. So here, acetylcholinesterase is an enzyme. It's located in that synaptic cleft, which breaks down now acetylcholine. And when it breaks down acetylcholine, it's going to break down acetylcholine into acetic acid and into choline. Into acetic acid and into choline. So acetylcholine's effects are going to be terminated now. Its effects are terminated as soon as acetylcholinesterase breaks it down. So breaks it down in that synaptic cleft. Also, what happens to acetylcholine is acetylcholine can diffuse away from the junction as well once it's done whatever it needs to do. Right? If it stays bound to that receptor, it's going to keep triggering signals over and over again. So we can't have that happen. So acetylcholinesterase comes in, helps breaks it down. Once it's done, all right, cool, get out of the way, get done, picture's over, finish, and let's move on. So this removal of acetylcholine, what it does, it prevents continued and 100% guaranteed, most likely undesirable muscle fiber contractions in the absence of additional nerve stimulation. So this removal of acetylcholine prevents continued muscle fiber contractions in the absence of additional nerve stimulation. There's many toxins, drugs, and even diseases that can interfere. So there's many toxins, many drugs, and even diseases that can interfere with the events at the neuromuscular junction. One of those that I'd like you to know about is myasthenia gravis. Myasthenia gravis. Myasthenia gravis is characterized by a drooping upper eyelid. It's characterized by a drooping upper eyelid, kind of what I'm trying to mimic here. It's characterized by a drooping upper eyelid, also by difficulty swallowing, difficulty talking, and even generalized muscle weakness. And this is all due to a shortage of acetylcholine receptors. This is an autoimmune condition. And here what happens is patients, they make antibodies. They make antibodies to these acetylcholine receptors. So when you make antibodies to acetylcholine receptors, they're going to go and they're going to occupy these receptors. And when they occupy these receptors, acetylcholine can't bind. Now, if acetylcholine can't bind, acetylcholine then can't trigger the action that we should see at that muscle. So therefore, then that action can't occur. Right? The eyelid should be in its position, not drooping. Okay? So the acetylcholine binding is going to help to keep everything basically in place in relation to muscles. Next in here, we've got the end plate potential. Let's say we've got generated now. Let's move on to the generation of an action potential across that sarcolemma. So here we went through all these different terms. Okay? We saw these events. Let's look at these events then here through this video as well.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
So this will allow us then to move to the next section, which will be basically 9B. So here what we could do is close out of this screen and open up 9B. So here we've gone through and we've just mentioned these first parts in relation to slides 9B. So here we can see, now we talked about the destruction of acetylcholine, where we said acetylcholine's effects are going to be quickly terminated then by this enzyme. Now, we also mentioned acetylcholine is going to get broken down to acetic acid and choline. Okay, so I mentioned to you it's going to prevent continued muscle fiber contractions, right, in the absence of additional stimulation. Very important you understand, in the absence. Next in here, when we go through, we talk generation of an action potential across the sarcolemma, basically, is what we're going to see eventually as well. Now, I want you to know the resting sarcolemma. The resting sarcolemma, the plasma membrane, is said to be polarized, and it has voltage across its membrane. Okay, so when we talk about the resting sarcolemma, the resting plasma membrane, it's said to be polarized, and it has a voltage across its membrane. And also the inside is negative relative to the outside. Okay, so resting sarcolemma is said to be polarized, like we've seen before with the neuron. At the resting membrane, at the resting voltage, we said that the cell was polarized, right? That's why we were able to use certain terms that we used to describe the voltage when we described the cell depolarizing, or it repolarizing, or it hyperpolarizing, right? And so forth. And so, again, polarized at resting. It has voltage across the membrane. Now, here when we go through and we talk, again, the inside is going to be negative relative to the outside. And now an action potential. An action potential is going to be the result of a sequence of electrical changes. So an action potential is going to be the result of a sequence of electrical changes. It's going to be a result of a sequence of electrical changes. Now, once initiated, they're going to occur along the entire surface of the sarcolemma. So once the action potential is initiated, the action potential is going to occur along the entire sarcolemma. You'll see, and it's going to be a three-step process, and it'll be a three-step process. So you can see that here. It occurs in three steps. First, we're going to have a generation of an end plate potential, as you can see there. Second, we'll talk about depolarization, and then we'll talk repolarization there as well. So first here, let's talk generation of this end plate potential. So when we talk generation of an action potential, then across the sarcolemma, we've got to mention the first part, which is the generation of the end plate potential. And that's a local depolarization, okay? A local depolarization. Now, here when we go through, we see, I want you to know the binding of the acetylcholine. We talked about acetylcholine. Now, the binding of acetylcholine to the acetylcholine receptors at the neuromuscular junction, right? The binding of acetylcholine to the acetylcholine receptors at that neuromuscular junction, we saw it opened up chemically, or I told you ligand gated ion channels. So it opens up chemically gated or ligand gated ion channels that will allow sodium and potassium to pass. It will allow for simultaneous, okay, more specifically speaking, it will allow for simultaneous diffusion of sodium and potassium. Now, again, down there, concentration gradients. You should have that understood by now. I mean, we're 3 exams in. We've been talking about this since the first exam. So, diffusion of sodium is going to occur to the inside, right? Potassium is going to happen to the outside. We took the second exam. A lot of you guys got taken on that sodium potassium pump there as well. You have to pay attention to even the sodium potassium pump. I tell you guys a lot of times, the way they ask it will really confuse you guys. So, here we're going to see now a change in the membrane potential is going to occur as the interior of the sarcolemma becomes less negative. So more sodium is going to diffuse in. Now what's going to happen to the inside is the interior of the sarcolemma becomes less negative. So a change in membrane potential occurs as the interior of the sarcolemma becomes less negative. And that change you'll see is an event that we call basically local depolarization or an end plate potential. So again, since more sodium is diffusing in and making its way in, now the inside of the cell is becoming less and less negative, right? Again, no, what's going to happen is we're having, again, the word there is local depolarization. So I was going to give that word, but then I saw it's there again. So when this sodium is moving in, now let me not try to draw it in the air. Let's use this keypad I have here. So here, now let's say we were to draw out the voltage, okay? Now here we can see, now voltage, we're going to see basically on this side, and this side will have time. So here now when we go through, we saw initially, okay, we're talking resting. So we're at a resting voltage. So now if we're to continue, right, we'd be basically continuing with that line all the way across the screen. So here, let me put an M in front so that way it's more obvious for you. It's voltage, okay, millivolts. So here we're at basically resting voltage. So now here, if more sodium is diffusing in, like they're telling us right inside of here, more sodium is going to be diffusing in, the interior of the sarcolemma is going to become less negative, is what we're trying to understand here, right? So let's look at that. With more sodium diffusing in, now here you can see we're at resting. Now with more sodium making its way in, what's going to happen is we're going to see this voltage is going to start moving up, right? We have a local depolarization occurring. Let's say, just for example, I'm just going to throw a random number out. Let's say, for example, this is minus 90 millivolts. So here now it's moving towards, let's say, minus 60. So you see why it's becoming less negative? And now what's happening here is this is a local depolarization. There's a local depolarization, or now what we refer to as a EPP, I was saying, an end plate potential. An end plate potential. So a change in membrane potential occurs, I said, as the interior of the sarcolemma becomes less negative. Now the change is an event that's called a local depolarization. So initially that depolarization is a local event, and it's called an end plate potential. And it's called an end plate potential. Next in here we're going to see the actual phase of basically the generation and the propagation of the action potential, and we're going to check out depolarization part of the action potential now. So we've already generated the end plate potential. Okay, so let's we're going to go through. We're going to see. We're going to have this picture again. Okay, we went through and we saw this picture. Okay, so we're here in this region right inside of here. Okay, so we're creating those end plate potentials right inside of here. And now those end plate potentials are going to help to trigger basically that voltage change along this sarcolemma, where we're going to find, you can see here, voltage gated channels. So this is going to be like a domino effect. These dominoes, they start falling, and then what they're going to do eventually is they're going to hit the dominoes here on the side, and they're going to allow that, that whole domino cool effect kind of to make its way down. So here now we can see as we progress now, we're going to talk about generation of and propagation of an action potential. So the end plate potential, that local depolarization at the neuromuscular junction ignites. It ignites an action potential. It ignites an action potential by spreading to adjacent membrane areas, and I said by opening those voltage gated, we're going to see sodium channels. Because we're talking about the first phase of the action potential now, more specifically, right? Because if we're talking action potential, then we're talking the first phase, the second phase, and then even sometimes hyperpolarization, right? Depolarization, I should say, first phase, repolarization, second phase, and then hyperpolarization, the third phase. So now we're getting specific, so that's why I'm saying here that the end plate potential, this local depolarization at that neuromuscular junction, it ignites an action potential by spreading to adjacent membrane areas and opening up voltage-gated sodium channels there. That's what you're seeing. So here those end plate potentials are generated, and eventually now they're going to help to excite these receptors there, right? We've already gone through. We've looked at all the different events right at the neuromuscular junction. We're over it. That's so 15, 20 minutes ago, I think. Okay, so you saw that and how all that happened, right? You saw what was going on there. Simultaneous movement. You have to remember that simultaneous movement. So that's what we mentioned here as well. Right inside of here. They're not just using that word over and over for nothing, or nor am I. That's exactly what you saw in the picture too. You have to be able to describe that. I'm sure most of you guys have understood it in case if this happens to be an essay. Right? Again, a long process. I can make it start from talk to me about the generation, right, from this whole excitation, contraction part of it, right? From the excitation from the somatic motor neuron. So here you can see simultaneous diffusion. Okay, so let's move back. Come on, get me out of here. There you go. Eraser, erase this. Come on, this one's to erase, and let's arrow. Okay, so now here we're going to see sodium. Sodium enters. Sodium enters, and once a certain membrane voltage is reached, that certain membrane voltage is called threshold. So once threshold is reached, we'll see. So we could see sodium enters, and once a certain membrane voltage, we said called threshold, is reached, an action potential is going to be generated. Now, when we talk threshold, if you recall from before, it's about 15 to 25 millivolts more positive than what the resting voltage was there where we saw in our nervous, our neurons, our excitable cells. Now, ranges usually around minus 50 to minus 40 millivolts in most excitable cells. Now, when we talk in a typical skeletal muscle cell, you'll see the resting membrane voltage. The resting membrane voltage is going to be minus 90 millivolts. Minus 90 millivolts. In a neuron we saw, very good, minus 70 millivolts. Okay, just to give you an idea, epithelial cells, they describe as having a voltage of about minus 50 millivolts. Minus 50 millivolts. So here now, when we go through and we talk action potentials, the action potential propagates. The action potential propagates, meaning it moves along the sarcolemma. It moves along the length of that sarcolemma. And it's going to move along the length of sarcolemma, you'll see, in all directions. In all directions from that neuromuscular junction. So here you can see the action potential gets generated, allowing these action potentials now to propagate in all directions from that neuromuscular junction. As this action potential propagates, you can see this depolarization wave spreads to adjacent areas. It spreads to adjacent areas of the sarcolemma, and it opens up voltage-gated sodium channels there too. So as now these sodium channels are opening up, you can see this wave of this depolarization wave is passing, like we said, in all directions. So depolarization is happening, and you saw as depolarization occurs, then repolarization is going to follow. Now, like depolarization, repolarization is a consequence of changes in membrane permeability. It's a consequence of changes in membrane permeability. Now, in this case, sodium channels close and the voltage-gated potassium channels open. The sodium channels close and the voltage-gated potassium channels are going to open. Now, since the potassium ion concentration is higher inside the cell than in the extracellular fluid, potassium is going to diffuse out, as we can see in this bottom picture right here. So potassium diffuses out of this muscle fiber, and when it diffuses out of the muscle fiber, what it's going to do is it's going to help restore that negative charge, right? Because potassium has a positive charge to it, as we saw when we were talking action potentials before in relation to neurons. So when we start to kick out now this positive charge, let me see here. Okay, here we can see this in more of a descriptive fashion. So here you can see, number one, our local depolarization waves are going to be generated thanks to the simultaneous movement of sodium and potassium. An end plate potential gets generated. So these end plate potential generations, you can see here, create this wave of depolarization that makes its way out of the synaptic cleft and onto these nearby voltage-gated channels, right? They're voltage-gated channels. So what this wave of depolarization is, it's the voltage change. So this voltage change is the signal that the voltage-sensitive channels need. Does that make sense? I hope that makes sense. So, I mean, that's convenient. Look how convenient. They two happen to be right next to each other. And we are very well engineered. So here you can see now that wave of depolarization is going to spread to this adjacent area, now activating these voltage-gated sodium channels, so triggering them open, and then helping to generate that action potential. So the action potential then is going to be propagated, we said, along this sarcolemma. So the action potential itself now is made up of, first you've got this depolarization phase when we're talking from resting. So let me see if there's an actual graphic image there. Otherwise, I'll go back to the picture we were at and draw a graphic there and put the two things together. Okay. Oh, very good. Okay, so let's do this. I want to actually put this and this together. So I'm going to put this same picture here, right? This action potential, okay? Resting voltage, Okay, you can see minus 90, minus 95 millivolts is showing here. And then here we're going to have depolarization. And what happens there? Sodium channels open. Okay, so then you have this upstroke, and that's all due to sodium's entry. Sodium making its way into the cell, right? Sodium has a positive charge, so the positive charge making its way into the cell. Now the cell's inside is minus 95. Now positive charge making its way, and that's why it's going to become more positive. And then sodium channels are going to close, potassium channels open, kicking potassium out, okay, is where we were at, and then helping to restore that charge back down to resting. So that's the repolarization phase. So I'm going to put this picture right here into this image here. So here now, was it, yeah, this image, or let's use, let me see. Let's use this image here. I like this image. So here again, let's draw. That was horrible. Let's erase that. I wish it wasn't so flimsy. Okay, so here, this is the hard part. Okay, and there. So here now we're at resting. So again, we're at resting. Resting we saw about minus 90, minus 95 millivolts. Okay, and now we're going to have basically this end plate potential come in. The end plate potential moves now. Again, remember we're checking this voltage we're looking at is going to be here on the sarcolemma. It's not voltage right inside of here. Okay, we're looking at voltage over here. So here now we're going to see, now we're at minus 90 to minus 95 millivolts we said, right? So here, let me write that in as well. Minus 90, what was that? Okay. So minus 90 minus 90 minus 95, that's irrelevant really right now. Here's where we are at. So now what we're going to have is, you can see we're at resting. Now, as soon as this depolarization wave starts to spread off to this adjacent area here, you can see we said these sodium voltage-gated channels will open. Here you can see when we talk voltage-gated channels, remember we described even what voltage-gated channels look like. Now here you can see they're getting descriptive. They're showing you the activation gates and then the inactivation gate, versus in the potassium, we saw we only had one type of gate. So here you can see now, sodium starts to rush in now because the voltage changes. So here you can see that's being depicted now also by these negative signs on the outside versus the positive signs on the inside, where we are supposed to have negative signs on the inside and positive signs on the outside. So here now the voltage changes occurred. When the voltage change occurs, it opens up, we said, these sodium voltage gated channels. Now what that's going to do is it's going to cause this then upstroke here. So the upstroke is going to be basically what we're seeing here in this part of that action potential. So that happens due to the influx of sodium. Okay, if we're just monitoring this area right inside of here. And then eventually what's going to happen is you can see this area next to this adjacent area here now is going to have its voltage changed as well, you can see. And that again, there is that domino effect you see spreading to adjacent areas. So that will trigger then, if we had sodium voltage gated channels here, that would pop open now because of the voltage change. Okay, so let's look back at one. So now here, this voltage change now in this membrane is going to cause now these potassium channels to open. These potassium channels, they're going to open now when the voltage becomes, you can see, positive. So that triggers them to start opening. So now when they start opening, here what happens is we said that those sodium channels close, and now here we're going to move from depolarization into repolarization, where now our potassium channels open. So here the potassium channels open, you can see sodium channels close, we said, right? Potassium channels open. So we move down to this picture here. We're now in repolarization. So you can see that right inside of here, right? This is repolarization. They've even mentioned it right down inside of here for you. And then here we saw depolarization. Okay, so now here in repolarization, okay, Potassium channels are open. Potassium channels are open. Potassium carries a positive charge. Now potassium is allowed to now leave. So potassium channels open. When that opens, potassium is going to diffuse. And we've seen, normally speaking, potassium is going to be found in higher quantities on the inside. So when it diffuses, it moves from, again, then higher concentration to lower concentration. So it moves out. When it moves out, you can see that positive charge now is going to be lost that we had here, and now restoring that cell's inner negative charge. And you can see inside now, we've got negative charge restored. So this wave of repolarization is following that wave of depolarization. As that wave of depolarization is moving, you can see in its direction. So potassium diffuses out, and the muscle fiber restores then that negative charge. Now, during repolarization, a muscle fiber is said to be in a refractory period. During repolarization, a muscle fiber is said to be in a refractory period because the cell cannot be stimulated, again until repolarization is complete. So let's look at then excitation contraction coupling. Now, when we talk excitation contraction coupling, it talks about the sequence of events by which transmission of an action potential along the sarcolemma leads to the sliding of the myofilaments that take place inside of the cell. You will see the electrical signal. That action potential does not directly act on the myofilaments, right? Instead, you saw before we covered, it causes a rise in intracellular calcium ions, which allow those filaments then to slide. So here you can see they're telling us that as well. The events that transmit action potentials along the sarcolemma are going to lead to the sliding of those myofilaments. The action potential is brief, and it ends before the contraction, you can see. It ends before the actual contraction. So the electrical event first, and then the actual physical event. Right? The action potential, that electrical event, is going to cause a rise in intracellular calcium. That makes sense now, right? That's why the action potential is first, and then it ends before the contraction, because it causes a rise in the intracellular calcium, which then is going to lead to the actual contraction occurring. And then we have a latent period. This is time when that excitation contraction coupling event occurs. And this is the time between the action potential's initiation and the beginning of contraction. So that's your latent period. That's a time between the action potential initiation and the beginning of contraction. So let's go through and let's check out now all these events. We said the action potential is going to be propagated along the sarcomere. It gets propagated along the sarcomere, and then you have the T-tubule there, so it gets propagated along the sarcomere and then dips down into the T-tubules. And then there we said we had voltage-sensitive proteins. These voltage-sensitive proteins are going to stimulate calcium's release from that sarcoplasmic reticulum. right? And then I mean, this I'm just summarizing it because you saw there's actual voltage gated channels there that you had to know the name of, and then you have these foot protein type channels that we said you had the name of there as well, and then is the actual calcium behind the terminal cisternae. So here I'm just saying, oh, the voltage sensitive proteins stimulate calcium's release, but what's actually involved with all of that. Okay, and that calcium is what's necessary for contraction. We said it's going to provide the final go for contraction. So here we can see now all of these events. Now we've got our motor neuron. Okay, here we can see our somatic motor neuron forming the neuromuscular junction. Again, another neuromuscular junction we can see there. So these are all the terminal branches. Right? We said we can have very many terminal branches coming from one axon. So here we're looking at one neuromuscular junction. So let's zoom in on this and we'll look at all these different events basically, and we'll put this is all excitation, contraction, coupling basically together. So here let's move over and let's look at now this neuromuscular junction in greater detail. And when we look at this neuromuscular junction in greater detail, you can see there, summarizing the events, we saw the action potential making its way down. I was going to summarize it, but let's go through it in detail. We saw the action potential making its way down the axon terminal, right, helping to open up voltage sensitive calcium channels. Voltage sensitive calcium channels open up, allowing calcium influx. Calcium influx leads to the binding, calcium binds to proteins, leading to then the exocytoses of these synaptic vesicles. These synaptic vesicles contain acetylcholine, so they release acetylcholine into the synaptic cleft. The acetylcholine binds to the postsynaptic membrane receptors, which are ligand-gated receptors. So you can see how we're going from voltage to ligand to voltage. So you have to be able to explain these different receptors you're seeing there. So now these these ligand-gated receptors there get basically excited, allowing simultaneous movement of sodium and potassium and helping to trigger, we said, end plate potentials. These end plate potentials now are going to then cause the voltage in the neighboring membrane region to then open up. So here you can see that action potential is going to be now basically spread across in both directions. So now these voltage gated channels open up here, and the sodium voltage gated channels open up on this side, same thing on this side, basically in all the different sides, allowing then that voltage wave, that deep voltage basically change, that depolarization wave to be propagated along the sarcolemma. And we said then we have these T tubules, which we said were invaginations of the sarcolemma, and how convenient. So they allow this depolarization wave, this action potentials to travel down. And here we happen to have voltage sensitive channels there as well. Now these voltage sensitive tubule proteins we've got there, okay, you can see in greater detail, will open up and they change shape. Basically they move, and when they move, they're no longer pushing up against, you can see these other receptor proteins. So when they're no longer pushing up against these other receptor proteins, here what happens is calcium then is allowed to make its way out. Allowing calcium to make its way out. So here now we can see when calcium then floods the cell, calcium is going to go in and calcium is going to bind, right? This takes us to then cross-bridge cycling, muscle fiber contraction now, right? So calcium then makes its way out, and calcium goes and binds to troponin. Troponin, we saw, is going to move that tropomyosin, which is initially blocking the active sites on the actin, moves the tropomyosin off of those binding sites for myosin, exposing those binding sites, ready for myosin, and myosin then forms cross bridges, and we have contraction occurring. So let's check this out.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
All right. So here we can see now basically these events, okay, all coupled together. Now, channels involved in initiating muscle contraction we saw there as well. I told you make sure you're paying attention. Now, here you can see nerve impulses are going to reach the axon terminals where voltage-gated calcium channels open. Okay, then acetylcholine gets released into that cleft. Now, once acetylcholine gets released into the cleft, then we're going to see here acetylcholine binds to its receptors on the sarcolemma, and this is going to open basically these ligand-gated channels. Okay, and then the end plate potential occurs, opening up voltage-gated channels. So you see we're going voltage, chemical, voltage, and then here you can see we have voltage-sensitive proteins on the T-tubules. Okay, so those first channels on the T-tubules, the voltage-sensitive, I gave you guys the names the other day as well, are going to be your DHP, dihydropyridine. Okay, dihydropyridine proteins. These DHP proteins you have there then will change shape, then allowing then basically our foot protein that you have underneath them, the ryanodine proteins, to move out of the way, and not ryanodine, RYANODINE is the proper spelling, and allowing basically the calcium to flood the cell. Now, role of calcium in concentration. So at lower intercellular calcium concentrations, tropomyosin is going to block the active sites on actin. Okay, the myosin heads cannot attach to the actin, and the muscle fiber is considered to be relaxed there. Now, when we have higher intracellular calcium concentrations, calcium binds to that troponin, as we said. The troponin changes shape, and it moves the tropomyosin away from those myosin binding sites. Myosin heads bind to the actin, causing the sarcomere to start shortening, right? We said the H zones are going to come together. The H zone disappears, the I bands and the Z discs come together. And so you have basically shortening occurring. Now, when nerve stimulation ceases, the calcium gets pumped back into the sarcoplasmic reticulum and contraction is going to end. So here then what we could do is we could move over to then 9C, and here we can see now cross-bridge cycling. All right, so cross-bridge cycling is what we're talking here. We can see now, we talk cross-bridge cycling, it's going to continue as long as calcium is there and also adequate ATP, as long as the calcium signal and adequate ATP is present. So as we've seen basically all these different events, okay, contraction is going to continue as long as the calcium signal and adequate ATP are going to be present. So cross-bridge formation, okay, high energy myosin head attaches to the thin filaments. Okay, that's the actual cross bridge. It's like a ratchet, we said. So now then you have the power stroke, the working power, basically the working power of the power stroke take place. That's the when this ratchet actually does the job. So that's the stroke part. Now you can see the myosin head, it pivots and it pulls. And it's going to pull the thin filament towards the M line, right? We saw the M line is found in the H zone. So now that's why we said the H zone disappears, because now this continued contraction, right? Attachment, detachment, or attachment, pulling, detachment, attachment, pulling, detachment, attachment, pulling, detachment is going to bring those thin filaments towards the M line. So we'll get to look at that here as well. Now, cross-bridge detachment. Cross-bridge detachment, I told you, ATP. ATP is going to be required. So ATP attaches to the myosin head and cross-bridge detaches. Now the cocking of the myosin heads is going to occur because of the energy from the hydrolysis of the ATP. So the hydrolysis of ATP cocks the myosin head into a high energy state and gets it ready for cross-bridge formation, for attachment and cross-bridge formation. So here we could see that. Here we have now ATP. ATP has come in. We've had now ATP come in. ATP comes in, and ATP is there. Calcium is in the picture. Calcium helps to expose those binding sites now on the thin filaments, the myosin binding sites on the thin filaments, by rolling the triple myosin over. So here now you can see the myosin heads are able to go and attach now to those myosin binding sites on those actin filaments. Now cross-bridge formation occurs, and now what's going to happen is you can see the power stroke takes place next. So here we move down to the second picture. Here we have the power stroke occurring. Now this is the actual working stroke we said, or the power stroke. Now here is where you can see we're going to pull the thin filament in. So now if we're looking up in this picture, right, this is the picture we're zooming in on. That's what we're looking at. Now when you look at this is the actual thick filament we're looking at, and then here we can see the thin filaments right on top. So now this is happening on the top, this is happening on the bottom, and it's happening on all different sides. Same thing is happening on this side here. That's why we said these thin filaments are going to be brought in towards that M line, which is right here. So attachment, detachment is bringing in this side here and bringing in this side here. That's why you see these arrows here as well. That's why the H zone will disappear. The I bands and the Z discs you see here are going to move in closer together. They won't be so spaced out like we saw in that picture where we actually looked at both the relaxed state and the actual contracted state of muscle. So here now what we could do is we could move forward, and we move forward, we can see now once the power stroke occurs, we bring this thin filament in closer now on both sides. This ADP we had attached, you can see, now is basically used up. It's gone now. So here what happens is we said we need ATP for the detachment. So now ATP comes in. When ATP comes in, now ATP comes in and it attaches to the myosin. Now the link between the myosin and actin weakens. It weakens, and it causes the myosin heads then to detach, causing then that cross bridge now to break. Causing the cross bridge to break. So here now you can see we needed ATP for the detachment there as well. So now this ATP, it undergoes hydrolysis. It undergoes hydrolysis, and you could say is hydrolyzed. And when it gets hydrolyzed to ADP and this inorganic phosphate. And now it's in the cocked position. As soon as calcium is there, and if the sites are exposed, cross-bridge formation will continue. Like we said, it will continue as long as calcium signal and adequate ATP are present. Power stroke. And then the detachment. Now there's a condition called rigor mortis. When we don't have ATP anymore, and that happens after we die. Now, this is in the process of decomposition. Here, what happens is the ATP that we have gets used for the detachment of muscles, and there you'll see this person then is going to be in this contracted state, but now we don't have that ATP for the detachment. So this muscle then will stay in this contracted state as this process of decomposition occurs. You'll see then it'll start to basically soften up from this stiffened state. And that'll be all then thanks to decomposition. So the ATP, we don't have that problem occur, but as soon as we run out of that ATP, then, obviously that happens in after death, and so that's, again, in the process of decomposition we described. And so you can see there then, ATP comes in and allows myosin heads to become, they described as being cocked or in a higher energy position. So let's check this out.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots. sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
All right. So rigor mortis, we can see there as well. Crossbred detachment requires ATP, and that ATP is not there. Happens about 3 to 4 hours after death, to get a little bit more specific. And you can see then the body, 3 to 4 hours after death, you'll see it begins to stiffen, and then weak rigidity at 12 hours post-mortem, basically there. So here we can see now the dying cells, they start taking in calcium, and then cross-bridge formation starts to occur, right? And then there's no ATP that's generated to help break those cross bridges. Now, when we move forward, we'll talk about the contraction of skeletal muscle. I want you to know there are two main categories of contractions. Isotonic and isometric contractions. Now, when we go through and we talk, muscle changes, you'll see muscle is going to change in length. And when muscle changes in length, right, we said when it shortens, muscle contracts. Now, we're going to see muscle is not only going to generate force, basically, here by shortening, but we're also going to see it's going to generate force also by lengthening as well. So we'll go through and we'll check all that out. But here, muscle is going to change in length, okay, and it moves a load. And you'll see thin filaments are going to slide. Now here we'll look at, again, isotonic and isometric contractions. So here we go through and we talk isotonic contractions. Here, before we actually jump into isotonic and isometric contractions, I want you to first understand two terms. I want you to first understand one term, and it's called muscle tension. Muscle tension. When we talk muscle tension is going to be the force exerted by a contracting muscle. It is the force exerted by a contracting muscle on an object. The force exerted by a contracting muscle on an object. And the second term is load. Load is the opposing force. It's the opposing force exerted on the muscle by the weight of an object that's being moved. So load is the opposing force exerted on the muscle by the weight of the object being moved. So now let's look at then isotonic contractions. First, isotonic contractions, are going to be contractions where muscle length changes. Isotonic contractions are contractions where muscle length changes, and moves a load. There are two types of, we can see here, there are two types of isotonic contractions. We have first what we call concentric contractions. First we have what we call concentric contractions, and second we have eccentric contractions. Now when we talk concentric contractions are contractions where muscle shortens. Where muscle, you can see here as well, muscle shortens and does work. Where the muscle will actually shorten and it does work. Such as if you're picking up a book. Okay? The muscle will shorten and you do work. Or if you're kicking a ball, okay, muscles will shorten and you'll actually do work. Now, when we talk eccentric contractions, now eccentric contractions are contractions where muscle generates a force as it lengthens. Muscle generates a force as it lengthens. For example, your calf muscles when you're walking uphill. Like your calf muscles when you're walking uphill. Here you can see an example of concentric isotonic contractions where you can see now this muscle is going to shorten and it lifts a load, it moves a load. So on stimulation, this muscle, it develops enough tension, okay, enough force, you can say, to lift the load, to lift that weight. Once the resistance is overcome, the muscle shortens, right? And the tension is going to remain constant for the rest of that contraction. Next, we have isometric contractions. So here you can see an isotonic contractions as well. Here you can see now the tension that actually gets developed. You have zero tension in the muscle to actually having, let's say, about 3 kilograms of tension in the muscle. Now, here what happens is as this tension goes up, you can see the length, the muscle length decreases. So the muscle shortens, and the muscle shortens, you can see in line with as soon as we hit full-on resistance there. And you've got the actual job then being done. The weight was being moved there until then the muscle relaxes, and then you can see the muscle will then go back to its resting length. So next thing we have isometric contractions. When we talk isometric contractions are basically where tension may build in the muscle, okay, where tension may build in the muscle, but the muscle neither shortens nor lengthens. Where tension may build in the muscle, but the muscle neither shortens nor lengthens. Isometric contractions occur when a muscle attempts to move a load greater than the force the muscle is able to develop. So you'll see cross bridges are going to generate force, but do not move actin filaments. So the muscle neither shortens nor lengthens. This occurs when a muscle attempts to move a load that's greater than the force that the muscle is able to develop, like trying to move a piano or trying to move a refrigerator. So here you can see the length of that muscle is going to remain the same. Tension gets generated, but we said, so neither shortens nor lengthens, but you don't move anything. In class, I would move the table, that I'm at, and give you an example there of the table, a fridge, or piano, like we mentioned there as well. Oh, you can actually see me. I forgot. Yeah, the table, I would have right there in front of me. I would actually, try to lift that, and you can't lift that table. So you can see there, the muscles are actually generating that force, and you can see they're not shortening nor lengthening, okay? And it's not enough to move whatever object you're trying to move. Yeah, I forgot you're here. So here you can see that being illustrated now the same way our isotonic contractions were being illustrated. The muscle is not shortening nor lengthening, and nor is it able to actually do work. So let's move then to muscle metabolism. Let's talk muscle metabolism. Now, providing energy for contractions. Okay, ATP supplies energy for cross-bridge formation. Okay, want you to understand that ATP supplies energy for cross-bridge formation, for detachment, and also for the operation of calcium pumps in the sarcoplasmic reticulum. And for the return of sodium potassium after excitation contraction coupling occurs. Right? Sodium potassium ATPase pump. Now, when we go through and we talk ATP, muscle stores very limited reserves of ATP. Muscle stores very limited reserves of ATP. Muscle stores about 4 to 6 seconds worth at the most. Just enough to get you going into a physical activity you may be doing. Now, once broken down, ATP is regenerated, and it's regenerated by one of these three pathways. ATP is regenerated by one of these three different pathways you see here. Either you have a phosphorylation of ADP, adenosine diphosphate, right? If we phosphorylate ADP, ATP is created. So if we have a direct phosphorylation of ADP, that happens thanks to creatine phosphate. Happens thanks to creatine phosphate, and the enzyme that will allow this to occur is creatine kinase. Creatine kinase. I believe we have a picture here. We can see that right inside of here. So here we have direct phosphorylation. Here in direct phosphorylation, okay, here you can see now ADP is phosphorylated thanks to creatine phosphate. Creatine phosphate then becomes, you can see here, creatine. Or we can have ATP being regenerated by, so that's why you see here creatine is used, you'll see by athletes as well, involved with helping to basically regenerate ATP for them and so forth. So here you can see now we have another way of regenerating ATP, and that's going to be thanks to anaerobic glycolysis. Thanks to anaerobic glycolysis. Anaerobic glycolysis converts. It converts glucose to lactic acid. It converts glucose to lactic acid. Okay, glycolysis itself does not require oxygen. You saw that. So anaerobic glycolysis, where we convert glucose to lactic acid. And then third, aerobic respiration. Aerobic respiration. Okay, so anaerobic pathways, you can see 2 ATP is the net gain there and a whole bunch of lactic acid. And then aerobic pathways, it's a more proper way of forming the energy. 95% of it, you can see. And then you guys know everything there as well. We've gone through aerobic pathways and we've checked them out fully in the beginning of the semester. Right? All these different pathways. You see, that picture doesn't look so scary now. So all three pathways we can see there. creatine phosphate. Creatine phosphate is a unique high energy molecule that's stored in muscle. It's a unique high energy molecule that's stored in muscles, and it's used to regenerate ATP. Okay, so we saw that basically there. Together, stored ATP and creatine phosphate are going to provide power for about 15 seconds. Provides power for about 15 seconds. So here you can see now that, so the first 6 seconds we said is ATP that's stored in muscles. It's going to get used up first to kind of get us going with the activity if we're doing short duration exercise. And then the next 10 seconds you can see there, okay, to give us, like we said, about 15 seconds, 16 seconds of energy. ATP is formed from creatine phosphate and basically ADP. So that's through direct phosphorylation. And then after that, you can see we'll basically start jumping into glycogen that's stored in muscles, and it gets broken down into glucose, and that's going to start generating ATP for us. And by the end of exercise, right, anaerobic pathways have kicked in, and that's why exercise is going to end. But if we can do some prolonged energy there, prolonged exercise, that prolonged duration exercise, hours of exercise, ATP is going to be generated, you can see, by the breakdown of several nutrient energy fuels by aerobic pathways. And so glucose, okay, glycogen isn't going to be the only two. That's when then we start jumping into protein breakdown even, and lipid breakdown there as well. So several nutrient energy fuels are going to kick in and ensure then, and as long as we're breathing, right? As long as we're providing that oxygen, that's what they'll tell you when you're doing exercise as well. Make sure you're breathing, whether you're running or whether you're lifting weights, that oxygen there is going to be very important. So let's talk then velocity and duration of contraction now, basically. Here we're going to be talking about muscle fiber types now as well. So I want you guys to know muscles are going to vary in how fast they can contract and in how long they can continue to contract before they fatigue. These characteristics are going to be influenced by muscle fiber type and by the load. So let's talk first muscle fiber type. I mean, load is obviously going to be, respective to whatever the load may be there. So let's talk muscle fiber type. Now, when we talk muscle fiber type, there are several ways of classifying muscle fibers. Okay, but learning about these classes will be easier if you pay attention to just two major functional characteristics. If you pay attention to just two major functional characteristics. Number one is the speed of contraction. The speed of contraction. When we talk speed of contraction, we're talking the basis of speed of shortening. We're looking at the basis of the speed of shortening. There are two types of fibers. The difference reflects how fast their myosin split ATP. First, you have what we call slow fibers, which are going to split that ATP slower, and then you have the fast fibers. So the basis of speed of shortening, there are two types of fibers here. The difference reflects how fast their myosin split ATP. So slow fibers and fast fibers. You can see both right inside of here. So slow fibers of the speed at which myosin ATPase is going to split ATP. Next then, we're going to look at the major pathway for forming ATP. The major pathway for forming ATP. Now, the cells that rely mostly on oxygen-using aerobic pathways are oxidative fibers. Again, the cells that rely mostly on the oxygen-using aerobic pathways, those are oxidative fibers. And those that rely more on anaerobic glycolysis, Excuse me. So again, and those that rely more on anaerobic glycolysis are our glycolytic fibers. So again, the cells that rely mostly on oxygen using aerobic pathways are going to be our oxidative fibers. And then when we talk fibers that rely mostly on anaerobic glycolysis, they will be our glycolytic fibers. So let's look at then all fiber types. 3 types now. We have slow oxidative fibers and fast oxidative fibers. And then we have fast glycolytic fibers. Most muscles contain a mixture of fiber types. So you can see, since they have a mixture of these different fiber types, the range of contractile speed, fatigue, and resistance is going to be, again, be able to be altered there. So you have a range in regards to speed, a range in regards to fatigue, and a range then in regards to resistance there as well. Now again, you can see all fibers in one motor unit are the same type. And genetics dictates individuals' percentage of each. Okay? And then obviously your environmental factors, you actually doing exercise or using them or not utilizing them is going to be another factor, right? Arnold Schwarzenegger's kids have these genes that will not just in his kids directly, but eventually in his lineage, that will help in with this increase in percentage of certain types of muscle fibers. And again, if they use them, then they can have something like that there as well, a body that would look similar to what his is. It doesn't mean just because you're his kids, you can have bodies like that. No, no, no. So here now, when we go through and we talk different fiber types, so here we can see slow oxidative fibers. Now we talk slow oxidative fibers. So we said first there, we saw slow fibers, fast fibers, again, depending on how fast they're going to split their ATP. Okay. So we have then slow oxidative fibers. So when we talk slow oxidative fibers, you can go through this list here and you can understand speed of contraction is slow, myosin ATPase activity is slow, and now here the primary pathway for ATP synthesis is going to be aerobic pathways, and myoglobin content is high, glycogen stores are going to be low, and now you can see recruitment order, rate of fatigue there as well. So recruitment order, these are going to be first to be recruited, and then when you talk the fatigue, these are going to be slow. They're fatigue resistant fibers basically here. So when you talk these slow oxidative fibers, they're endurance type basically fibers or for activities of endurance type activities. Example, running marathons or maintaining posture there as well. And when you look at the structural characteristics, then here you can go through and look at the structural characteristics there. So here then we have fast oxidative fibers. Now we talk fast oxidative fibers here, speed of contraction fast. Okay, so you can go through the list and you can get an understanding there. Now they're best suited for sprinting and walking. And then our fast glycolytic fibers. We talk fast glycolytic fibers. Again, you can go through the list and get the idea down. And these are for short-term or basically short-term intense or powerful movements. So various fiber types. Next in here we have muscular dystrophy. We want to talk about, basically when we talk muscular dystrophy, it's a group of inherited muscle destroying diseases. And they generally appear in childhood. And here what will happen is muscles will enlarge. Muscles are going to enlarge like we talked about with Duchenne's muscular dystrophy. And muscles will enlarge due to fat and connective tissue deposits. And muscle fibers are also going to atrophy and degenerate in these patients as well. Duchenne's muscular dystrophy, okay, most common and severe type, it's inherited, sex-linked, it's carried by females, however, it's expressed only in males, it's seen only in males. And we talked about you having the patient having lack of dystrophin there. So 1 in 3,500 percent, basically, or 1 in 3,500 cases there. Now, when you look at it in greater detail, you'll see here the cytoplasmic protein that stabilizes sarcolemma is going to be lacking in that patient, as we described yesterday in the last class. So you have a fragile sarcolemma that tears, calcium entry, okay, is going to increase. That leads to damaged contractile fibers, leading to inflammatory response and inflammatory cells, leading to then muscle mass dropping because of the inflammatory cells infiltrating that region. And then the victims are going to become clumsy and they fall frequently, and usually they die of respiratory failure in their 20s. Fortunately, no cure. Prednisone, you may have heard of, steroid. It improves muscle strength and function. Again, what it does, it helps to reduce the inflammation, which then helps to improve muscle strength and function. Okay. So here you can see viral gene therapy and infusion of stem cells with the correct dystrophin gene is showing some promise. So, stem cell research and obviously gene therapy are very promising in certain diseases, hopefully with everything, but it's just a positive thinking, I believe, from my end. All right, that's where we'll end. So we end muscles.